Hello and welcome to all my students. I hope you all are doing fine and must be studying well at home. So now, today we are going to continue with the chapter Cell the Unit of Life, where we are going to deal in with the detailed structure of a prokaryotic cell. I just hope you all have gone through the first lecture in which we have introduced cell and the types of cells on the basis of nucleus, that is prokaryote and eukaryote. Now from there itself we are going to continue further, and now let's try to understand the detailed structure of a prokaryotic cell. First, starting with the cell envelope, right? Okay, so here goes the first heading, and now the first heading is going to be structure of prokaryotic cell. Structure of prokaryotic cell. And first we are going to discuss about the cell envelope. That is the surrounding of the cell. Now in case of prokaryotic cell beta, cell envelope is made up of 3 layers. Outer to inner, they are glycocalyx, cell wall and cell membrane. Each layer has some definite function to perform, but overall all these 3 layers together, they act as a protective unit. Correct? So what is the first point that you are going to write over here? Cell envelope beta helps in providing protection to the cell. First point. Second, consists of 3 layers, which are from outer to inner. The 3 layers are beta glycocalyx, followed by cell wall, and the innermost being cell membrane. This we have arranged from outer to inner. Correct? Now, one by one, we are going to discuss about each layer in detail. First, starting from glycocalyx. Right? Now, first point, you yourself knows what ma'am is going to write, of course, that it is the outermost layer. Correct? Yes, absolutely correct. So, being the outermost layer, one of the most important function that it has to perform is protecting the underlying layers, right? So, the most important function of glycocalyx is going to be protection, right? Now, the most important thing which I am going to tell you about glycocalyx is, beta, depending upon the nature, the material of which it is made up of, glycocalyx can be of 2 types. Either it can be present in a very slimy form, mucilaginous form, sticky form, or it can be present in the form of a thick, tough, resistant layer. So if glycocalyx is present in the form of mucilaginous slimy form, then it is known as slime layer. And if it is present in the form of thick, tough, resistant layer, then beta glycocalyx is called as capsule. Right? Now, slimy layer function, of course, it is protection. But apart from protection, if I talk about specifically, slime layer also protects the bacteria from getting dehydrated. That means it avoids the excessive loss of water from the cell. However, if glycocalyx is present in the form of capsule, then it protects the host immune system. It protects the cell from the host immune system. Why? Because the capsulated bacteria is not getting recognized by the host system. As a result, it persists in the system and multiplies and yes, causes disease, so on. Correct? So this glycocalyx beta can be present in two forms, either in the form of loose sheath, mucilaginous means sticky, right? Then that is known as slime layer, correct? However, if it is present in the form of thick, tough, resistant layer, beta, then it is known as capsule. Very, very important question can be asked in your NEET examination related to slime layer and capsule. Now just write their individual functions also. So the individual functions of slime layer, what did ma'am tell you? Yes, slime layer beta, if this is present, if glycocalyx is present in the form of slime layer, it protects the cell from dehydration. It protects the cell from dehydration. Correct? Yes. And what is the second form? The second form is capsule. So for capsule, you're going to write protects from host immune system. Protects from host immune system. Correct? Got my point? This much better you have to do for glycocalyx. Now followed by glycocalyx, the next layer inner to glycocalyx is cell wall. So first point that you're going to write for cell wall is it is present inner to glycocalyx, right? Followed by second point, it is a dead layer, non-living layer, rigid layer, permeable layer which does not act as a barrier for the movement of materials in and out of the cell. Correct? So cell wall is basically dead, non-living, thick, tough, resistant. Correct? So basically it is a dead layer and is a permeable layer which does not provide barrier for the movement of material. Correct? Understood everyone? First point, second point and third point. Now, moving towards the fourth point, and let's try to understand that what this cell wall is made up of. So when question comes that bacterial cell wall is made up of which chemical, then your answer will be very good everyone. Peptidoglycan, also called as murin. So next point that you're going to write over here is the cell wall is made up of murin. The other name for murin that is more common is peptidoglycan. Now, murin, peptidoglycan, can you just decode this word for us? Because we are not able to understand that what actually cell wall is made up of. We only know certain macromolecules like lipids, proteins, carbohydrate, everything. But what is this peptidoglycan? Can you decode for us? Yes, of course we can decode. Now this peptidoglycan, try to just break the word itself beta. Peptido, right? And glycan. Peptido word, peptide. Peptide means protein. Protein means amino acid. Glycan, something related to glucose. Glucose means sugar. So this peptidoglycan is actually a combination of different amino acid and sugar units. Right? So what is murin? Murin is actually peptidoglycan which is made up of protein and sugar. There are 4 amino acid and 2 polysaccharide which joins together with the help of the bond to form murin. The 4 amino acids are that makes up one peptidoglycan is L form of alanine, D form of alanine, L form of lysine, and D form of glutamic acid. Whereas the two complex sugar that makes up the peptidoglycan are NAG. You all know NAG beta stands for N-acetyl glucosamine. Whereas NAM stands for N-acetyl muramic acid. Correct? So these 6 components, they bind together with the help of the bond to form one unit of peptidoglycan. Like this, so many units of peptidoglycan are present, which again cross-link to form the cell wall. Right? Now, beta, there is one antibiotic called penicillin. This antibiotic, penicillin, does not allow the cross-linking of peptidoglycan, or even if cross-linking is existing, then in the presence of penicillin, that cross-linking gets destroyed. As a result, cell wall synthesis does not take place in the presence of the antibiotic penicillin. So that is why penicillin kills bacteria by degenerating or by degrading their cell wall. How? Because penicillin inhibits the cross-linking between the peptidoglycan units. Right? Got my point? This is a special note point which is very important for you to know. NEET or in your 12th, 11th examination, question related to this can be asked, that which antibiotic will attack the cell wall and how. So both the questions, now I hope they are answered. If yes, then just write it down. Antibiotic penicillin beta inhibits the cross-linking of peptidoglycan units. Right? As a result, cell wall synthesis does not take place. As a result, cell wall synthesis does not take place in its presence. Right? Clear? Chalo. Now moving with the function of cell wall. So the most important function of the cell wall, if it is asked, then it is that to maintain the shape of the cell and avoiding the cell from bursting. If cell wall is there, then no matter whatever be the amount of water, it is going to adjust inside it and will protect the cell from bursting. If you remember, there was a question that if you keep a RBC, human animal cell, inside the hypotonic solution, and parallelly you keep a plant cell also inside a hypotonic solution, so which among them will burst and why? So animal cell, that is RBC, will burst due to excessive endosmosis. However, plant cell, even due to endosmosis, it is not going to burst because somehow cell wall will prevent the bursting by adjusting the excess amount of water. And now since animal cells are not having that cell wall, so RBC due to excessive endosmosis will burst. So the main function of the cell wall is protecting the cell from bursting. Right? Protecting the cell from bursting, right? Now, the second is that maintaining the shape of the cell. Third, provides strength and rigidity. Correct? So these are some important functions of the cell wall. I hope the cell wall is also clear to everyone. Now proceeding with the third and the innermost layer of the cell envelope, that is cell membrane. Now unlike that of beta cell wall, cell membrane is living layer. It is selectively permeable. Selectively permeable means will allow selected material to move in and out of the cell. Correct? Got my point? Now, protection is one of the function of the cell membrane also, as it is a part of cell envelope. But apart from protection, it helps in bulk transportation of material, being owing to its selective nature. Right? Clear? Shall we write these points? Chalo, come on. Write these points along with me, beta. So the innermost layer is now cell membrane. The other name is plasma membrane, you know. The first point you're going to write is innermost layer. Second point you're going to write is it is a living layer. Third, it is selectively permeable, right? So what is the nature of this cell? It is basically innermost. Second, living. Third, it is selectively permeable in nature, right? Now, as I was telling you about the function will be explained in a better way also when I'll teach you the detailed structure of plasma membrane. In that, we will also talk about the function. But here, just write the name itself, just write the function. Explanation will be done later on. Correct? So, basically, it helps in bulk transportation of materials. Bulk transportation of material. Now talking about the structural organization and what this plasma membrane is made up of. So if you all remember that plasma membrane is made up of two macromolecule. One lipid, second protein majorly. Right? Now how this lipid is arranged, how the proteins are arranged, everything ma'am will teach you, but not over here. So here you just have to simply write one line, write it and proceed further. When the right time will come, there's a complete plasma membrane model in your syllabus that has to be taught, but not now. So here just write structurally, beta, plasma membrane is made up of, it's made up of what? It is made up of lipid bilayer plus protein. This model was explained by Singer and Nicholson in the year 1972. So when I'll teach you the structure and completely, there we will talk about this model as well, right? Now, you have seen one thing, beta, that unlike that of cell wall, plasma membrane is living in nature. Living means it can show certain modification. Accordingly, it can change its function. It can give rise to new structures. Living का तो यही मतलब हुआ ना. Living से तो समझ में आया कि चलो इससे हम और भी काम करवा सकते हैं. Like for example, if a layer is dead, so apart from providing mechanical strength and support and rigidity, no other function you can expect from a dead layer. But if it is a living layer, then you can expect more functions. You can modify the layer according to your wish. to perform more and more functions, right? So same thing happens over here with the prokaryotic plasma membrane being living. It can perform so many more functions by modifying itself. So now I am going to teach you further modifications of plasma membrane in prokaryotic cell. So the next heading is modification of plasma membrane. Now this modification I can divide into two categories depending upon the location, that whether the modification is inside the cell or whether modification is taking place outside the cell, matlab whether plasma membrane is going outward or whether plasma membrane is projecting inward. Accordingly, we have two categories of modification, right? So inner modification, in which you have to just study about one. Yes, you all guessed it right, mesosomes. Whereas when it comes to outer modification, beta, we have three category as pili, fimbriae and flagella, right? So how many modifications you have to study? 4. 1, 2, 3, 4. Right? Chalo. First, let's start with mesosomes itself. As I told you that this is the inner modification of plasma membrane. So what happens over here? Beta, plasma membrane is going to infold. It is going to fold on the inner side of the cell, either in the form of vesicle, like vesicle, you know, cup-like structure, or in the form of tube, or in the form of branched network like lamella, right? So this is your plasma membrane which has got infolded either in the form of vesicle or in the form of tube or in the form of lamella. All these three representations are actually mesosomes. All these three representations are nothing but your mesosomes. Correct? So plasma membrane, basically when it infolds, it increases the surface area. Now why? Because beta, these infoldings of plasma membrane, so basically mesosomes are nothing, just the infoldings of plasma membrane that contains respiratory enzyme, right? So these vesicles or tubes or tubules, beta, they contain, yes, they basically contain respiratory enzymes. That means they behave as mitochondria. And mitochondria, if you remember, yesterday I was telling you that in prokaryote cells, membrane-bound organelles are absent. That means if they don't have membrane-bound organelle, that means they don't have mitochondria, they don't have powerhouse of the cell. So who is going to synthesize energy for them? Do they don't require ATP for their metabolic activities? No, they do require ATP. However, instead of having mitochondria, the same function in them, in prokaryotic cell, is beta performed by these mesosomes. ये जो mesosomes है ना, ये वही काम करती हैं जो माइटोकॉन्ड्रिया यूकैरियोटिक सेल में करता है. Why? Because these infoldings have enzymes, respiratory enzyme that helps in the breakdown of glucose to produce energy. Correct? And now these enzymes should function in a better way. For that they need more surface area, As a result, plasma membrane enfolds inward. Got my point, everyone? Yes or no? So what are mesosomes? These are the inner foldings. These are the inner modifications of plasma membrane analogous to eukaryotic mitochondria. Why? Because they contain respiratory enzyme, hence they can help in respiration. Got my point? So what will you write over here? For mesosomes function. First function. They are beta analogous means same function but different origin. You all know homologous, analogous. Analogous means same function. Now, mesosomes also perform the same function as mitochondria. Both helps in glucose breakdown, but both have different origin. One is originating from the infolding of plasma membrane and second have some other origin. So analogous to eukaryotic mitochondria, as they help in respiration. Correct? Got my point, everyone? Now, apart from helping in respiration, beta, they also help in DNA replication and they also help in binary fission. So, if you remember the process of binary fission, now what happens in binary fission? Suppose this is my bacterial cell which wants to undergo division, and this is the genetic material. So, this plasma membrane is having infoldings. These infoldings are nothing but the, yes, mesosomes. So now whenever this bacteria wants to divide by fission method, these opposite side infolding beta, they start growing towards each other like this. They start growing towards each other. Now while they grow towards each other, they give the signal to genetic material. This is the DNA, to undergo replication. That means apart from performing respiration and division, they also help in DNA replication. So give signal to DNA replication, and then a time comes when both the mesosomes, they finally join each other, thus dividing the bacteria into two. So mesosomes, they not only beta help in respiration, but they also help in fission. Plus, they also help in DNA replication. So these are the different functions of bacteria. Sorry, mesosomes. Correct? Got my point, everyone? Chalo. Now proceeding further, this is the inner enfolding of plasma membrane. Similarly, you have three more modifications of plasma membrane which are the outgrowth. That means they are growing outside the cell, not inside. Correct? If the outgrowth is in the form of hair-like structure, then this is known as pili. This is my plasma membrane. If it is in the form of bristles, you all know what are bristles? You all must do brush now. So the part of the brush, toothbrush, where bristles are present, number of hair-like outgrowth but coming in the form of tuft, coming in the form of gucha. So that is called as bristle-like. So this bristle-like arrangement is known as fimbriae. And the third is, if this modification is in the form of long thread-like, then this thread-like outer growth of plasma membrane is known as flagella. Correct? Yes or no? So how many outgrowths do we have? Pili, fimbriae, flagella. Now always remember beta, pili is made up of pillin protein. Fimbriae are made up of fimbrine protein, and flagella, yes, is made up of flagellin protein. The main function of flagella is helping in locomotion, right? Yes or no? So those bacteria who have flagella, they are motile, they can move. But those who lack flagella, they are sessile, they cannot move. Correct? Talking about the second, that is fimbriae. Fimbriae beta, the main function is helping in attachment of bacteria to the substratum. Suppose rock. Now there's a bacteria who wants to attach to the rock. Rock is the substratum for the bacteria. So who's going to help the bacteria to have a good grip on the rock? Fimbriae, right? Whereas pili helps in exchange of genetic material. Helps in exchange of genetic material. Correct? Yes or no, everyone? So these are the three outgrowths. Number 1, pili. Number 2, fimbriae. Number 3, flagella. Where pili helps in exchange of genetic material, fimbriae helps in the attachment, and flagella provides locomotion. So these are the outgrowth and mesosome is the ingrowth. I hope all the modifications of plasma membrane are also well understood and clear to each and every student. Now, proceeding further, this is about the cell envelope. Proceeding further with the inside the cell envelope, what are the extra structures found? So beta, cell envelope encloses the fluid which I told you yesterday, cytoplasm, right? The cytoplasm actually act as the main arena where all cellular activities takes place. The cytoplasm is rich in non-membrane bound cell organelles and some storage bodies which make the metabolic functions of prokaryotic cell easier. So now cell envelope is done, which consists of how many layers? 3 layers. Outer to inner, glycocalyx, cell wall, cell membrane. Glycocalyx, protection, and it is the outermost layer which can be either present in two forms, either in the form of mucilage or in the form of thick tough resistant layer. Then we talked about cell wall, where I told you the composition of cell wall, the function of cell wall, and that it is dead in nature. Then we came to the innermost layer, that is plasma membrane, which is unlike that of cell wall, is a living layer, selectively permeable in nature. Main function is transportation, and then we discussed the modifications. Till here, I just hope everything is crystal clear to everyone. One topic I have left, that is detailed structure of plasma membrane, which I will teach you, but once eukaryotic cell starts, not over here. Right? Done? Now let's proceed further with the composition which is present inside the cell envelope. So here we are now going to deal in with, right everyone, cell envelope encloses the fluid, and that fluid is known as, yes, very good, cytoplasm. The cytoplasm, NCERT says, it is the main arena for all cellular activities. right? It is the main arena for all cellular activity, first point. Now, what does cytoplasm consist of? So, beta, the cytoplasm, I'm only talking about prokaryotic cytoplasm, not eukaryotic cytoplasm. So, when I talk about prokaryotic cytoplasm, it consists of non-membrane bound cell organelles called ribosome and dead bodies, storage bodies called inclusion bodies. Correct? Yes or no, everyone? I hope you all have little bit idea about ribosomes. So what are ribosomes? First point, non-membrane bound. Non-membrane means it lacks any defined membrane around itself. Non-membrane bound cell organelle, which is also known as protein factory. Correct? Now, why is it known as protein factory? Of course, because it helps in protein synthesis. It helps in protein synthesis. Correct? More details we are going to do about ribosomes. So, let's start and let's move to the next page. See, these ribosomes, beta, they are granular structures. When you view them under the microscope now, in the cytoplasm, you will see some granular structures. So there was a scientist called George Palade who was the first one to report these granular structures as ribosomes. Right? Now, in case of prokaryotic cell, there are two locations where you can find ribosome. One, you all know, they lie free in the cytoplasm. Second, they can also be found attached to the plasma membrane. Correct? Next point. The average size of a ribosome is approximately 15 into 20 nanometers square. Correct? Now, what these ribosomes are actually made up of? So, these ribosomes basically, beta, they are made up of protein plus one of the component of RNA, that is rRNA, ribosomal ribonucleic acid. Correct? Got my point? Shall we write these extra points which I have told you so far? Chalo, write it. So ribosomes are granular. Granular means like, you know, like crystals, like salt crystal, they're granular, small particle form, right? So these are granular structure, non-membrane bound structure found either free in the cytoplasm or attached to the plasma membrane. Or attached to the plasma membrane. Next point, beta, they were discovered by George Palade, whose average size is 15 into 20 nanometers square. Right? Clear? Understood, everyone? I just hope you all have understood this much very clearly. If yes, then let's continue further. Now, see beta, ribosomes are granular structure. They are found free in the cytoplasm or attached to the plasma membrane. You know, they were discovered by George Palade and their average size is approximately 15 into 20 nanometers square. Now, when I talk about the function of ribosome beta, then their main function is helping in protein synthesis. That's why they are known as protein factories. इसी कारण से these ribosomes are known as protein factories. Right? Clear? Now, are they capable of synthesizing ribosomes alone, or they need the help of some or the other structure? So as I told you, alone beta they cannot synthesize protein. However, they need the help of another type of RNA called mRNA, messenger RNA. Correct? So they protein synthesize. So there's mRNA chain on which number of ribosomes attach. So these are the ribosome. Now together they help in protein synthesis, and this simple structure is known as polysome. You can also call it as polyribosome. Correct? This is very, very important. You should know this thing. And what these ribosomes are made up of? So these ribosomes are basically themselves beta made up of proteins plus rRNA. Right? Done? I hope you all have understood so far. Now beta, there are two types of ribosome. ज़्यादा तो नहीं बताऊंगी types के बारे में, but little bit information I will give you over here, and remaining information we will do when I will teach you eukaryotic cell in detail. But since we are doing prokaryotic cell, little bit information about prokaryotic ribosome, you should know it. So basically beta, there are two types of ribosome. How many types of ribosome? Two types. The two types of ribosome are 70S and 80S. 80S are exclusively found only in eukaryotic cell. Right? So these are exclusively 80S type of ribosome is exclusively found in eukaryotic cell, whereas 70S type of ribosome is found in prokaryote. This ribosome is beta made up of further two units, a larger unit and a smaller unit. Correct? Now, this larger unit is actually called as 50S and smaller is known as 30S. So this is my smaller unit on which there's a larger unit, and this is my 70S subunit. 30, 50 together making 70. Now you might be thinking कि अरे यार मैम का मैथ्स खराब हो गया है क्या? Ma'am does not know mathematics. Ma'am, you are telling 50 plus 30 70. अरे मैम 50 plus 30 makes 80. How are you telling 70? So students, मैम का मैथ्स नहीं खराब हुआ है. Actually, the case over here is you don't have to add the units. It's not like 50 plus 30 making 70. No, these are not the additive units. You simply, or these units or these numbers are basically calculated on the basis of their density gradient. So if 30S is called as 30, then it's because of this density. If 50s is called as 50, then it's because of its density. And if together when they are placed, if the density is coming 70, so accordingly the name is 70s. So all these numbers, they are not additive, rather it is based on their density gradient, which is calculated on the basis of the amount of protein and rRNA found inside them. You all know that ribosomes are made up of rRNA and protein. Ma'am told just now that these units, they are made up of proteins and rRNA. Now, there are different types of proteins, there are different types of rRNA. Every protein, every rRNA has their own density according to their mass and volume. So, like this, density is calculated and accordingly these values come. Right? Correct? Got my point, everyone? Similarly, beta, this 80s is also made up of two units. The larger is 60s and smaller is 40s. Again, you don't have to add, otherwise it will become 100s. You simply, this is again just on the basis of their density gradient. So you need not to add these values. Right? Clear? Chalo. Now, question arises, ma'am, okay, you told that these numbers are on the basis of density gradient. But what about this S? S is the unit which tells you about the density gradient, correct? Like you have centimeter, you have meter, you have millimeter, liter, decibel, all these are different units now of different quantities. So similarly, for ribosomal unit, the unit is S, that stands for sedimentation coefficient. Sedimentation coefficient, which gives the measure of the density and sizes, which gives the indirect measure of ribosomal units density and sizes. Units density and sizes, right? Apart from this, it was discovered by Swedberg. So S can also stand for Swedberg's unit, right? Clear? So either you call it a sedimentation coefficient or after the name of the scientist, Swedberg unit. Got my point? I hope ribosome is absolutely crystal clear to everyone. Now, apart from ribosome, beta, they also have some dead storing bodies called inclusion bodies. These inclusion bodies are also non-membrane bound. Like, you know, in every home, you keep a dustbin now. Whatever waste is generated or whatever unwanted product is there in your home, so temporarily you keep it in the dustbin and then in the morning you throw it out. Similarly, these inclusion bodies are like dustbins for the cell, where cell stores all the reserve material, excess, unwanted secretory products temporarily. Right? Clear? So now next heading you are going to write is inclusion bodies, which are also widely present in the cytoplasm of prokaryotic cell. So these are also beta non-membrane bound storing bodies. These are also non-membrane bound storing bodies found freely in the cytoplasm, right? Clear? Now, what is their function? Function is storing. What storing? Storing any kind of reserve material, like for example, phosphate granules are responsible for storing phosphate. Sulfur granules, they are responsible for storing sulfur. Then cyanophycean granules, etc. Right? Clear? Now, one more example I would like to quote over here, beta. And that example is gas vacuole. These are also the inclusion bodies, also called as pseudo vacuole, which are actually responsible for storing gas. So these gas vacuoles, also called as pseudo vacuoles, they are basically responsible for storing gas, thus providing buoyancy to the cell so that it can simply float on the surface of water. They are restricted to cyanobacteria and some purple green sulfur bacteria. So you have to remember, they store gas, thus providing buoyancy. I hope you all know buoyancy, Archimedes principle. Yes, and they're exclusively found in blue-green algae, which is actually cyanobacteria, a type of prokaryotic organism. And second is purple green sulfur bacteria. So these are the prokaryotic cells that are found floating on the surface of water owing to the presence of gas vacuole, which is one of the non-membrane bound inclusion body responsible for storing gas. Now, since they store gas, it's just like balloon. एक balloon है, उसमें हवा भर दो, हल्का हो जाएगा ना, it will start floating. And now if you remove that air, it won't get that buoyancy effect, so it won't float. Similar to this also. So when, if inside any cell, if I fill air, it is going to become lighter in weight and it will start floating. So same thing is done with the help of this gas vacuole, right? Clear? I hope till here all the points are absolutely done and dusted. So with this, the entire structure of prokaryotic cell is done. Now let's try to draw the diagram of a typical prokaryotic cell depending upon the information which we have collected so far, right? So now we are going to draw the diagram of a prokaryotic cell. Now, starting with the envelope, right? So, a prokaryotic cell is beta surrounded by how many layers of cell envelope? 3. Outermost being glycocalyx, cell wall, cell membrane. So, how many layers should I draw over here? 3. Now, this glycocalyx can be either present in the form of mucilage or can be present in the form of a thick layer. If thick layer, then I'll draw a complete line. But if I want to show mucilage layer, then I draw it in the form of a dotted manner, right? This is just the representation, nothing extra. So suppose if I am taking my glycocalyx as mucilaginous layer, then hence I will draw this layer in the form of dotted manner, showing its mucilaginous property. Followed by thick, tough, resistant, rigid layer, that is cell wall, and then the innermost plasma membrane. Now this plasma membrane, beta, is it present continuous or some foldings are there? Of course foldings are there. So folding can be either in the form of vesicle or tube or lamella. Right? Clear? So this is the outermost layer, glycocalyx, where it is present in the form of slime layer. This is cell wall, and here goes your innermost layer, cell membrane. All these three together provide protection and is commonly called as cell envelope. Right? Yes or no? Have you all understood so far? Chalo. Very good. Now beta, this infolding consists of respiratory enzyme. So these infoldings are nothing but mesosomes that contain respiratory enzymes. Now, the fluid which is present inside this envelope, this fluid is known as cytoplasm, correct? Which encloses double-stranded single circular DNA that is called as nucleoid or genophore. Apart from this, they also have extra genetic material I told you yesterday by the name plasmid. Right? Then this cytoplasm is also rich in non-membrane bound cell organelle found either free in the cytoplasm or attached to the membrane called as ribosomes. ठीक है? फिर along with ribosomes beta, they also have some other non-membrane bound storing bodies by the name inclusion bodies. Very good. Followed by more outgrowths of plasma membrane, like for example, locomotory organ, flagella. They may also have tuft of, guच्छे में, hair-like outgrowth called as fimbriae, plus they can also have some more hair-like outgrowth called pili. Now, check the whole diagram. This is the typical diagram of a prokaryotic cell. Everything which map, okay, one more, ribosomes, polyribosomes also you can draw. You know what are polyribosomes? So this mRNA along with number of ribosomes makes the polyribosome that helps in beta protein synthesis. Correct? So this is the whole diagram of a well-labeled diagram of a prokaryotic cell, which now you have studied after studying the detailed structure of the cell. That's it for prokaryotic cell. I hope you all have well understood it and no one is having any doubt related to it, right? If you have any doubts, feel free to post in the comment section. And you know, you are getting these lectures freely on the YouTube channel PW English as well as on the app. So if you're not able to download the app and if you're not getting the access or whatever is the trouble, don't worry, they are freely available on the YouTube channel as well. Go through them, watch them, and if you have any doubts, any queries, feel free to post them on the comment section. Right? Yes or no? So that's it for for prokaryotic cell. Now we are going to start with the eukaryotic cell. Beta, typically there are two broad categories of eukaryotic cell, plants and animals, right? So first of all, you should know the basic difference between plant and animal cell. Somewhat idea you all must have been having, if I'm not wrong. Many of you must be having an idea of plant and animal cell, the basic difference. Let's do few over here. So divide your notebooks into two columns as plant cell and animal cell. So first point of difference is beta plant cells are comparatively larger in size as compared to animal cells. So you will write that they are smaller in size. First point of difference. Second point of difference is that in plant cell, vacuoles are centrally placed. Vacuoles are large and well developed, whereas in animal cells, vacuoles are poorly developed or even absent sometimes, right? Third point. Now you know who needs more energy, plant or animal? किसको ज़्यादा एनर्जी चाहिए? कौन बातें करता है, वॉक करता है, टॉक करता है, खाना खाता है? All these activities are performed by us, and for all these activities we need energy, and for energy we need mitochondria. So in short, animal cells are having more number of mitochondria, and here the number of mitochondria are less. Next, plant cells have a unique feature of cell wall being present, but the cell wall is absolutely absent when it comes to animal cell. Animal cells, in them, the only outer limiting layer is plasma membrane. They don't have cell wall. Correct? Next, there's one special cell organelle called plastids, which are found in plant cell but absent in animal cell. Similarly, centrosome is one of the cell organelle which is found in animal cell. Centrosome is a cell organelle present. Here it is absent. Correct? Yes or no? So these are the basic point of difference between plant and animal cell of eukaryotic cell. Now that's it for today. The details of eukaryotic cell, the structural organization and everything we are going to do in our next class. In our next class, we will cover the cell wall structure, cell membrane structure in detail, as well as we are going to cover the endomembrane system also in next class, right? And then further in the next class, we'll cover semi-autonomous cell organelles, structure of cilia, flagella, and we'll try to cover more portions because I think we just have 4 classes for this chapter, and we'll try to cover them up, right? So I hope you all have understood. Now, moving towards some questions. How many layers are found in a cell envelope of bacteria? Yes, answer is 3. Next, extra chromosomal material of prokaryotic cell, yes, is known as plasmid. So I hope you all have understood the entire lecture which has been taught to you. If you have any query, as ma'am always says, post in the comment section. I'm going to reply. Thank you for watching me. Till then, bye-bye everyone. Take care. Study well, keep working and get success.